Hello, everyone. My name is Daniel Rubianto, and I am a Wildlife and Fisheries Resources major here at West Virginia University. Today, I will be presenting to you my project that I've conducted with the help of my research mentor, Dr. Christopher Rhoda, on the difference in the number of complex songs by songbirds between a rural and urban environment. Before we get started, I wanted to give some background information to help give a better understanding about my project. As the world continues to modernize, the population of human beings also continues to increase. This increase in the human population also leads to an increase in urban noise, such as noises from cars and construction, for example. For this study, we focused primarily on songbirds, or more specifically, the northern cardinal, song sparrow, and white-throated sparrow. These three songbird species rely heavily on vocals for attracting mates and establishing territory, which makes these species victim to high levels of urban noise. Our questions for this study were if increased urban noise would lead to a change in songbird phonology and pattern, and if there was a change, if it would affect the mating attraction success and/or territory acquisition that would influence population dynamics. Our purpose for this study was to find out if there was a significant difference in the number of complex songs by songbirds between a rural and urban environment. Our objectives for the study was to obtain the recordings of complex songs by songbirds between our two study sites, obtain the number of complex songs by target species for each recorded day, and compare the number of complex songs between our two sites. Moving on to our methods for this project, we started out with placing random points between our two study sites to deploy our autonomous recording units, also known as ARUs, which is what I will be referring to them as for the remainder of this presentation. We chose the WVU Core Arboretum as our urban study site, as it is in the center of Morgantown, West Virginia. We chose Priggett's Fort State Park as our rural study site due to the area's more secluded location. Our second step in our process was to deploy the ARUs at our points in step 1. We placed the ARUs 6 feet above the ground, strapped around a tree. The ARUs recorded sound from 4 hours before to 2 hours after sunrise. Our final process of the project was spectrogram analysis. In this step, we analyzed the spectrograms of the ARU recordings in 10-minute blocks at sunrise for each recorded day. For example, if sunrise for a recorded day was at 7:30 a.m., then the 10-minute block analysis would take place from 7:30 a.m. to 7:40 a.m. We would analyze the spectrograms for complete complex songs of our target species to count the number of complex songs for each species produced. The complex song was defined as any song with 2 or more notes produced by an individual. As you can see in figure 1, it shows a complex song of a northern cardinal that was present in one of our recordings. This is a great example of a complex song, as this spectrogram starts with 2 declining notes followed by several rising notes, all by one individual. Our results found that there was a greater song rate at Priggett's Fort, our rural study site, than at the Core Arboretum, our urban study site, as shown in the scatter plots for the northern cardinal and song sparrow. The white-throated sparrows were taken off our results as the complex songs were very rarely spotted on the recordings, with only one recorded day showing low counts of song signatures. We believe that our rural site had higher complex song counts across our target species due to a difference in urban background noise, as shown in the 2 spectrograms of our 2 study sites. There is a visible difference in the level of amplitude and frequency between the 2 sites. The Core Arboretum spectrogram shows much darker signatures, which means that it is much louder when compared to the Priggett's Fort spectrogram. This project has shown how an increase in urban noise can lead to a difference in the singing behavior of different songbird species. As the world continues to grow louder, it is important to recognize its impacts on wildlife species, such as songbirds who rely on their vocals for day-to-day survival. More research can be performed to follow up the study to further seek how other species may be influenced by higher levels of urban noise. I wanted to thank my mentor, Dr. Christopher Rhoda, for his guidance and assistance throughout this project, as well as the other undergraduate and graduate students in the lab for their help as well.